Hello!
Hi there.
Welcome to Microbiology Journal Club, where we know how big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. I would fact check for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated to the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist and currently working for a scientific journal.
Every week we meet to talk about microbiology, and today we're doing an overview. It's a news week. We do an overview of the coolest microbiology papers that we've seen during the week.
And at the end of the month, or it might be a bit later because we're working on this one paper, we'll be doing a deep dive on a paper that was selected by you, the public. The one we're doing next time around is going to be about BioInk, and we're very excited about it. But if you have any papers or any topics you want us to do a deep dive into, tweet us at MicroTWC and we'll take a look.
Absolutely. You can follow along with the papers that we cover on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at MicroTWJC. And boy, do we have a show for you today. We've got the infamous Delta-Omicron variants. Variants? Well, we're going to be delving in to see what that is all going to be about. What else do we have? We have, human coronavirus replication. So what is the coronavirus RNA during cellular infection? And also, we're going into a different virus, human cytomegalovirus, and the weird organelles it creates in the cells while it causes infection. But not only that, we've got an extinct tuberculosis infection from South America, and where that came from as a mystery. Unfortunately, I think I spoiled it in the title, but don't tell yourself that. We also got how do your intestines fight against helminths, against parasites, and how your gut bacteria can help that fight. Also, we've got identify how you can use a computer to identify drug-resistant bacteria by looking at how they're shaped, how round they are. We've also got a symbiosome. So, how do corals manage the bacteria that they partner with during their lifespan? And what does that mean for coral bleaching and other ecological catastrophes? We've also got E. coli population genomics, and finally we've got megaviruses. Marine jumbo bacteriophages. What are they about? So it's a jam-packed show, and I hope you'll stick around for all of the stuff.
Absolutely. And as always, I think we're open up with a bunch of SARS-CoV-2 papers. That's on everyone's mind. And this time, this term, Delta-Omicron, the internet loves smashing two words together and calling it a thing. In this case, we're talking about the potential for co-infection between these two strains and then a resulting recombination. And so these scientists will look at sequences to try to determine if that has actually happened.
Yes, this paper is called Evidence for SARS-CoV-2 Delta and Omicron co-infections and recombination. It comes from Helix, which is one of the companies who are doing a lot of testing in the US. And so they got all this testing data from when Omicron was first emerging and when Delta was just also there. So kind of at the point where there was a change of the guard between Delta and Omicron. So they thought that maybe in this section of the data, they'd find some crossover, some areas where you'd get more co-infections, because this was a time when both of these strains were sharking around and finding victims.
Right. And I feel like we picked out this paper, or you picked out this paper for us, because we had heard the news, right? Like that headline, Delta-Omicron, I saw it go around, and I was like, I don't know what this is all about. But it's nice to drive down and see maybe where those identifications would have happened, and what does it really mean?
Yeah, so I mean, Figure 1 paints the picture of what. So in 22nd, 28th November, all Delta, all the time. This is based in the US, so it hadn't jumped yet. And then throughout December, there comes that changeover, and that's where we see co-infections happening. So in the bottom graph, you've got the co-infections and the appearances of those. And then towards the end of that, they find a couple of recombinant viruses. So, I mean, it's interesting how they did this, because they've got these sequences, and the difficulty here is trying to figure out the difference between a co-infection and a full infection. So for a co-infection, you'd be looking for two whole genomes of the two different strains. So uh, and that's what they found for these samples. They found a mix between the two, but when you get a recombination, what you'd find is that you find like evidence of two different genomes, but the genomes aren't complete. So, for instance, you get the first part of the Delta genome, and then you get only the second part of the Omicron genome, and that's how the evidence they used to build up that there were recombinant variants coming around.
Right, and I think so they explain in the introduction here that the challenge of sifting out like the presence of recombinants here is like due to a bunch of technical factors in many ways, that the way that we sequence whole genomes, right, is like we have like all these little fragments. we're chopping up all the DNA that we get from a certain sample, and then we try to assemble them into these whole genomes. So there could be issues where, you know, maybe you had just like a little bit of something else, and that got chopped up in such a way that then those parts, like they're just contamination parts in some ways. They don't actually amount to a whole genome sitting inside of some individual. And then being able to, and then the other thing is that what if the mutations what if it's just the same strain, but it's picking up the same mutations? Or you have to be able to disambiguate those two circumstances.
Yeah, exactly. And it's quite a challenge. And yeah, well, and they believe that they found a couple of instances where there were these recombinant variants, but they only appeared in, I think, at select time points. So they didn't seem to spread very far. What they found was that based on the amount they saw these variants, they weren't actually. Because the thing about having a recombinant variant is that there's always a chance it's not going to be viable. Going to have it's like if you're trying to mix, say, a shark and a lion. They're both terrifying creatures, but sometimes you can mix them in a way to make them completely unterrifying. Like you got the top half of a lion and the bottom half of a shark, and it's just it's
gonna flop around. Yeah, it's gonna flop around in a strange way. But maybe we should briefly explain recombination. Is that like there has to be significant similarity for recombination to happen, and the idea is that during genome replication, right, the part of the genome is being made in a certain way, and then it sort of flips over onto the other genome, and then you get again that mermaid style. It's like, think of that on the genome level though, right? Like not so much on the whole body of the organism, but that would be the way in which recombination happens, right? You get one piece from another and another piece from a different strain.
Yeah, exactly. And because like sometimes when you've got an RNA polymerase, it's looking at things and you can like get it's an interesting process. So
I think one of the ways in which that they can find it, and this is one of the ways in which they'll disambiguate between, like, did the strain just pick up mutations that made it similar to the other, right, or not? They look for a breakpoint. So they're looking, because it's about reading the genome sort of in a linear fashion to pick up different pieces. You'll be able to say, oh, okay, well, all of a sudden now, all of the mutations on this linear genome are now coming from this other strain, and then it flips back. And so that's the evidence, I guess, that they're using to figure out if it's a recombination event versus just mutations being accumulated that are similar to one another.
Yeah. And from this paper, it does build up the idea that the recombination is something that can happen, but doesn't necessarily lead to a variant that is more viable than the others, because otherwise you would have seen some kind of spread during these initial spottings of the recombinant virus. And so, but the reason why it's in the news is not because of this paper, it's because the recombinant virus has been spotted in Europe, but unfortunately, most of that data comes from Twitter and GitHub. So so I've had to condense it using slides. So let me see if I can pull that up quickly. But yeah. And
Minraf says something in the comments, and I think I remember this in the news as well, that the first time we saw that name Deltacron in the news, like, it wasn't based necessarily on this paper that we're looking at, but rather some other ones that we're trying to find some definition. And I guess like again, what's really meaningful to people is it spreading throughout the population? And so like identification is just one part of that journey, and then you have to say like, is it going? And here, at least in the post hoc, the paper that we're looking at, it didn't seem like it went anywhere.
Right. However, this latest set of data comes from GitHub. So a strain of a fusion strain between Delta and Omicron was spotted by a PhD student working. So I've labeled the two people who were credited with discovering it on top of the slide. And so it was initially discovered in France, but subsequent versions of it have been discovered in Denmark, and they're calling it the XD variant. And based on what they've these sequences, it is a fusion of Delta and the spike from BA1. So so that I mean, that's interesting to me, because that doesn't sound like it should be a potentially threatening strain, because we already know that the spike from BA1 is being outcompeted by the spike from BA2 and Delta vice versa. And a lot of the research has been focused on the spike protein of these different coronaviruses. So if this is somehow like bucking the trend and actually surviving in the midst of BA2, there is a question as to what the rest of the coronavirus genome is doing, and what Delta was doing differently than the Omicron variants.
Which has been like a big, this is just the, this has been such a big question in terms of following the pandemic, right? That we only have a lot of detailed mechanistic information about the spike and maybe some of the NSPs we get dotted here and there, but like how a lot of the mutations impact the overall fitness and then how they work in combination, that's like so confusing to know, which I guess is the main takeaway from the story then of recombination, is that it can happen. It does happen, that there are people that are looking at lots of sequences and saying, okay, like here it is popping up. Will they be successful? Like that's sort of an open question still. And largely we haven't seen any successful recombinants moving around, but some do end up at least showing up and probably have some limited spread within the communities in which that they do appear.
Yeah, so at this point, it's quite hard to judge whether it can outcompete BA2. I mean, I'm of course hoping no, because this has appeared quite close to where I am, and rather not. I'd rather enjoy the sunlight a little bit longer. But
you could probably still enjoy the sunlight, right? Outdoors is like this good well ventilated outdoors activities. Those are good to
there's experiencing sunlight, and then there's enjoying it. And I feel like
okay,
I'm gonna. But yeah, this is so at the moment we're already in the middle of the BA2 variant spike, which is come at the worst possible time, because it's come at the time when everyone is trying to desperately end lockdown, and it's just everybody trying to keep safe as much as you can.
I wanted to pick out, again, I think this is like the challenge of trying to cover SARS-CoV-2 as it happens in real time, but like there was a bunch of Twitter threads that I saw that was talking about the BA2 spike, and I was like, okay. And I was trying to dive down to try to figure out what was underneath them, but I couldn't dig deep enough past Twitter to make some sort of cogent understanding of what they were talking about. It's just interesting because when we first talked about BA2, maybe last week or was it 2 weeks ago, there was the data coming from South Africa to say that the spike had gone up, and the Omicron spike was up and going down in South Africa. They had seen during that spike a huge increase in the proportion of BA2, but that didn't create like another bump necessarily. But it seems like what I'm seeing now from the various charts, right, is that that bump is being attributed to BA2, but I couldn't really figure out how that was being done.
So I think there are a couple of different factors that people are talking about on Twitter. I think firstly is prior vaccine immunity. That's a big thing. So the lack of it has been blamed for the big spike in Hong Kong. There's also a lifting of lockdown.
Sorry, I was going to say that's clear. To me, the Hong Kong case seems quite clear, right? That you can see that there's like a big discrepancy in the vaccination, like the elderly vaccination rate, thus leading to deaths.
Yeah, I mean, it's not just the vaccination rate, but it's also who you vaccinate. And by vaccinating the most vulnerable populations, like the elderly, like the immunocompromised, you're more likely to make a bigger dent on those mortality rates.
Right.
And then for other countries, it's the lifting of lockdown that is being attributed to the increase in the uptick in cases.
Right. Yeah, and so that's the point that I'm having difficulty disambiguating right now on Twitter. Like talking about Twitter as like the news source. Like, what's the contribution from the lifting of restrictions versus the contribution from the change in dominant Omicron variants?
Yeah, I mean, I should admit, I've been using lockdown in a very loose sense because I think a lot of these countries haven't been in actual real lockdown for like nearly 2 years. But
yeah, this is like
the idea of
restrictions, like kind of like mask restrictions. Yeah.
Yeah, restrictions and awareness and the acknowledgement that things aren't necessarily completely back to normal.
I also saw like increased mobility. What there was data showing that like people are just like moving around more now. Like that's been a big shift, and so that could also be driving this. But again, I couldn't disambiguate on Twitter.
Yeah, this part is there are a lot of unknowns, and that's the problem when we're living in the middle of something, and the answers only can come clear after we've had the chance to find out what the true answer is.
Right.
And so we'll find the answer long after it actually helps in any reasonable way. So the only thing we're left with is try and go with our best guesses and hope that we don't like end up with like Ghostbusters 2 when they avert the apocalypse at the end of Ghostbusters. They're a laughingstock in Ghostbusters 2 because once you prevent the apocalypse, people are like, did you really prevent the apocalypse? Or are you like that with that's like kind of where we are with the coronavirus outbreak. Where Ghostbusters 2, where everyone wants He-Man rather than Ray and Egon and all the rest to save them. So
all right. I think that's our 2 cents about this Delta-Omicron variant. People are keeping their eyes. Another takeaway is that there are people keeping their eyes on these things, and should we see the proportion of these recombinants grow, then I guess we'll definitely hear about it.
Yeah. And that moves us away from the SARS-CoV-2 coverage. So there wasn't much of this week. Still on the topic of coronaviruses, with the next paper, which is multicolor super resolution imaging to study human coronavirus RNA during cellular infection.
Oh, wait, did you want to talk about SARS-CoV-2 Omicron variant is more stable than the ancestral strain on various surfaces?
That is the second paper. I completely forgot about that. I skipped ahead.
It's a short one.
This is a very short paper.
It seems like they just they essentially take medium filled with Omicron and they sprinkle it over various surfaces, and then they have their other test case with the ancestral strain sprinkled over surfaces. Then they wipe it. They give that time. They just let it sit out, and then they wipe it up again, and they run that over tissue culture to get the TCID to figure out how many infectious particles are there. And the result that comes out of this one is that actually the Omicron variant seems like it stays around a little bit longer on these surfaces.
Yeah, I was quite excited about this when I first read it because I thought maybe this could be yet another like idea of how Omicron could work. But upon reading this paper, I just feel like there's a few things that are left a bit unanswered with it because we talked previously about the mechanisms for viral inactivation on surfaces, on fomites, and in the air. So there's the first thing about salts accumulating. So when SARS-CoV-2 gets expelled in a droplet and it dries out, the salts in that droplet start accumulating on the SARS-CoV-2 virus, making it less able to infect. And then there's the other thing, if it's from mucus, there are various things in the mucus that become much more sticky as things dry out, and that could also affect fomite contamination. And this paper doesn't really address either of those mechanisms.
No. I mean, it uses medium too, right? Yeah. So it's not even using like a physiologically relevant model droplet in that sense to gain this estimate.
So I feel like the medium they use, it should have like BSA and like salts in it, so that something of that range could be happening in it. The only thing you can take away is the relative differences. So this might imply that Omicron is more stable to salt to high salt environments or whatever like the most concentrated version of that medium is, might be more amenable to Omicron than the ancestral, and maybe that could be something to build on for further questions. But yeah, this is kind of very much a first step, and it'd be interesting to see what comes out from afterwards.
Yeah, but also like I was just thinking like I don't know how much of a difference like that would make necessarily, just because there was so much said about how it's a primarily aerosol like transmission route. So then if fomites is a very small percentage of the ways people get it, even if that like increased a little bit, right? Like any increased efficacy in that arena may not even result in like an overall transmissibility increase, was sort of my thought when I saw the paper.
Yeah, I think that is. So I guess this is why I kind of forgot about the paper, because it's fine.
I mean, the title is like, ooh, yeah, it's just fine.
It's fine. I think it's important that someone is asking this question, and I think it's a really interesting answer that I'd like to delve into a bit more, because maybe the amount it stays on fomite surface can also reflect on the amount it can stay active while airborne or other sorts of issues. So this can be useful in that respect. But yeah, my expectation of this paper I came in too high. I think I came in with too high expectations.
It's those titles. Those titles get you, right? Like,
yeah,
they're very exciting titles.
Yeah, that sounds like it's
okay.
Anyway, talking of ambitious titles, multicolor super resolution imaging to study the human coronavirus RNA during cellular infection.
I don't think we've talked too much about super resolution microscopy, but it is a fascinating technique. And I was looking up the one that they use in this particular paper, and I found some papers saying that it rivals SEM, in some cases EM. So typically when you're trying to look at really small things, you're limited by the wavelength of light itself, right? You can't look at anything smaller than the wavelength of light. So people go into electrons. People go into electrons to get a finer resolution. But a while back, I think it won a Nobel Prize, this technique was developed that can essentially look at the wiggling of like the sort of the statistical distribution wiggle of like that fluorophore, and then calculate exactly where it is. And so it was a big way of getting essentially a higher resolution from light images. And so using it in this circumstance, they're able to look specifically at the position of the RNA and the RNA and DNA, or I guess it's just
RNA
right?
and DS. So there's the guide RNA, and I think there's double-stranded RNA.
Yeah. Yes. Oh, yeah. Genomic.
Genomic. Yeah. Sorry.
Genomic RNA and the double-stranded. So, right, it's a positive stranded virus. So there's the genome itself, and then there's all the little pieces of the subgenomic RNA that gets made, and those things are going to then give rise to the proteins, right? And they're able to co-localize them and just see how that looks like in a single cell.
Yeah, I mean, it's interesting because I think we've like dipped into this where like coronavirus, when they infect cells, they create their own like what's been referred to as replication organelle, but they're basically vesicles which keeps the double-stranded RNA and single-stranded RNA safe from the immune system. Because even in cells, there are various proteins and enzymes walking around that are looking around for double-stranded RNA and trying to make the decision to destroy the cell if they spot it or not. So yeah, and here they actually find another way to visualize it, and because I think like double-stranded RNA is kind of indicative of like where replication is happening, where virions are being assembled, and single-stranded guide RNA is like where the RNA is being active. I keep saying guide RNA because my brain's hard of crisp bars. I mean genome RNA.
Yeah, I was gonna say it's hijacked our thinking of gDNA. It always is gonna be guide now because CRISPR is everything.
Yeah. Acronyms everywhere.
But yeah, essentially, I think what you said is a great summary. Being able to see where the double-stranded and where the genomic is giving a sense of like where these viruses are being made. And we've talked about this before, that viruses don't want to let their double-stranded DNA just hang out wherever, because then it can be seen by our innate immune system that says double-stranded RNA, like we don't make that sort of stuff, like something's going wrong here. And so being able to visualize that, I guess in real time, it seems like, right? Like these are living cells in which that they're taking these images of.
Yeah. That's another great thing about super resolution microscopy, because they are living cells, they're still alive. Whereas electron microscopy, you kind of have to freeze them, coat them in stuff, and they just don't. You get like a snapshot. Whereas this one, you can actually get a time frame, which is also quite cool.
Yeah. Well, yeah. Sorry, maybe living is not the right. They do fix them. I think.
Right.
At least it seems like they fix some of them, but like you can really quickly like fix and view it, instead of like covering up it with metals and things like that. So they're getting, it is snapshots, but the snapshots are like less processed. Yeah, that was my bad. I shouldn't have said living in that sense.
Oh, yeah. I jumped on that as well. I also made mistakes, so that's not.
But yeah, so you get these snapshots, and I guess it's mostly to show off the technology, being able to resolve these types of things, and also understanding that the double-stranded happens in like puncta, right? Like they're able to see like exactly like here is where the double-stranded genomic are together. It's a dot, right? And you can see like many of them across the whole surface. This is not just like it's not just a diffuse cloud. Like they can count if they needed to sort of figure out some sort of metric, and then they'll use that to, again, because it's largely a technology-based paper here, but they want to bring in like, how does this tell us anything about what's going on? They can then apply some sort of intervention. So they use remdesivir as a way of seeing like, how does that change the distribution, right, of genomic DNA associated with double-stranded DNA, and it does change that distribution in a certain way.
Yeah, I mean, it's quite interesting to give it changes things a little bit, because you don't really think of it like having like this broad application, but not the specific processes and the organization of them. And yeah, this paper is quite interesting into giving us that insight into the coronavirus life cycle. So that's why I found this paper quite cool. And yeah, I'm a sucker for good images. So
yeah, I mean, I think it's a science thing, right? Seeing is believing is kind of the statement, and then all this new microscope tech, you get to see like quite directly certain phenomena. The other thing that they notice is not just like the colocalization, but they look at the size of the puncta. So they're also saying that remdesivir like modifies the size of these colocalized areas in some way. And again, sort of the significance is to be determined, I think. But having this viewport is interesting.
Yeah, I mean, I should point out the thing they're experimenting on here is not SARS-CoV-2, but a related coronavirus called HCOV-229E, because I presume they don't have to have the safety issues with HCOV as well. And this is something they've been working on for a long time, so it's a lot more tools available.
Okay. And actually, this is like a segue into the next paper, in the sense that viruses need special spots to do their viral stuff. And in the next paper, they look at human cytomegalovirus, and they find that it is able to create a little partition, like almost a oil droplet style partition, a liquid partition inside of the cytoplasm. And again, that will have functional ramifications in terms of being able to isolate itself from maybe some sort of immune surveillance, but also it concentrates the factors needed to do whatever viral biochemistry is needed to be done. And so in this paper, they're investigating like how these, well, first of all, are these droplets what they think they are? And then how do they get formed?
Yeah. So, I mean, this is I'm quite interested in this because they're talking about like this thing called supramolecular condensates, which is a very complicated word for something that I think we do have an idea about. Because as you said, like oil droplets, it's like oil droplets, but they're protein separating. So we're talking about, I think, both things are types of proteins, but they behave in a different way that segregates from each other. So the way I tend to think about it is like the way that if you've got a box of cornflakes and all the small pieces go to the bottom, it's kind of like that, where where things of different sizes and shapes kind of like gravitate together. It's a bit more complicated than that because then I have to add in like, oh, but some of the cornflakes are magnetic in a certain and my analogies aren't always great, okay?
Yeah. No, but I like the visual. It's by their physical properties, right? Whether that be size or its affinity for other molecules, right? There's something that forces them together. And so like in the example of oil, that's hydrophobicity, right? But yeah, that's not necessarily the case in every condensate. And yeah, so they observe in infection that this virus does create sort of droplet-like looking things, and they are able to they have a hypothesis actually that they're created by a specific set of proteins that this virus makes, and so they put those proteins under, well, they make them glow with GFP, and they put them under control, inducible control, and then they have so they have then glowing bubbles that have this protein in them, and they're able to interrogate that whether or not they are liquid. So one of the ways they do it, they do a bunch of different things. I'll just highlight one of them because I thought it was interesting. They bleach like a little section of it, and they say, well, if it's liquid, then it should all kind of get mixed up again, and that little bleaching spot should disappear. And they see that happens. So I find that kind of an interesting way of verifying that these are liquid droplets.
Yeah, I mean, for those who try and watch the screen, I do apologize. There are some videos I'd like to show you, but because of the way this website displays them, it's all journal websites have some problems displaying videos, but this is quite an interesting one because I can give you a quarter of these videos rather than the usual full. Oh wait, let's see if I can do these full screen. Does it have a full screen option? Yes, it does. So yeah, these are looking over a certain set of hours and looking at how the various aspects of the cytomegalovirus associate within them. Yeah, they're not. Sorry, it really wasn't worth the effort. I'm sorry. They're not. They're not that.
It's just
not that great.
A little blip of yeah.
But apart from that, the actual research is great. The research is really interesting.
Yeah, it is. I mean, this is a very in-depth paper. They demonstrate that these are liquid, and then they go into this idea of is it the DNA? What is the nucleation point of these droplets? And they find out that the proteins that they think are making the droplets are also associated with DNA. And it's like the genome of the virus that's acting maybe as some sort of scaffold, attracting these proteins to get together, and then that sort of kicks off the making of these little bubbles, I guess, inside of the cytoplasm.
Yeah, that's quite interesting because it's you know how viruses they have these capsids around them they use to enclose their genetic material. It's kind of like that, but a lot more looser in function. It's almost like an alternative version of the virus that's only there to live inside the cell.
Yeah, right, because it's concentrating everything together. Yeah, I think that's really cool. Yeah, there's no membrane around these things, at least as far as they could tell. So yeah, that's also, kind of just interesting in terms of seeing these divisions. And again, we've talked, I think, a lot about this theme that cytoplasm is extremely structured. And even when something comes in and wants to hijack part of that cytoplasm, structure is important in making sure that the biochemistry that you want to happen is happening.
Yeah, I think that's all I've got to say on this paper. I mean, I'm always quite fascinated by this idea of biomolecular condensates and the idea that these viruses create these pseudo capsids inside cells and their own organelles,
Yes.
it's like they turn cells into virus cells, which is always a fascinating idea for me. Um, but yeah, moving on.
Okay. So now we're gonna move away from viruses and into more generally all sorts of different microbiology. This one is like a microbiology archaeology mix. Geographically dispersed zoonotic tuberculosis in pre-contact South American human populations.
So this is interesting because usually, like, I don't know if you ever read the book Guns, Germs, and Steel, but there's this idea from there that like there's a lot of like diseases that are associated with like the old world. So things like tuberculosis came from domestic animals. So in domestic animals, we've got this thing called in cows. There's like Mycobacterium bovis, which is like a cow version of TB. There's this idea that so therefore the idea that tuberculosis was present in South America in pre-Columbian times before like there was that exchange of diseases when Columbus went over to your country and messed things up. I
think the narrative on that one too is that like living in dense cities drives the evolution of these human pathogens, and then so those got introduced. But is that true really for the ancient history of tuberculosis? Because it is known that there's tons of different tuberculosis that infects all these different types of animals. And so, it's a kind of unclear whether or not we can just say, tuberculosis came from, these human evolved populations that then swept through the region, or maybe there's this alternate hypothesis where it came from a spillover event in a separate animal reservoir.
So what we're looking at here is in South America, there was they found like a bunch of like I think mummies or like dead bodies which had something that looked like tuberculosis, and where did it come from? Because it couldn't have come from that Columbian exchange, and so they looked into it and they found that the tuberculosis that was infecting these humans was related to Mycobacterium pinnipedi, which is a tuberculosis that infects seals. So
seal tuberculosis. So this isn't to say that like there wasn't tuberculosis that came during the Columbian exchange, just that there was another instance of it before that.
So this entire like version of tuberculosis infected this civilization and then went extinct before the Columbian exchange, which is a kind of a fascinating idea because we're looking at this outbreak that happened many like centuries ago, and for the disease that we have no idea about. And so I'm just kind of fascinated in that kind of the archaeology of it and the kind of problem solving of what was happening. I mean, if it's a zoonotic spillover, does that mean it was just people who were like hunting seals? Were they the only people infected, or did it spread like an epidemic?
Right. And again, like this is where like I watch this YouTube channel, PBS.
Yes, I
Beyond.
have.
You ever watched
great,
that one?
Graphics and visuals.
Yeah. And I feel like the thing that I really learned from there is that when you talk about stuff in the deep past, it's like you can tell a great story, but it's done with like so few data points that like as soon as like something new gets found, well, not as soon as, but as new things get found, you can revisit it, and the story will be incredibly different based on those new observations. And so it is important to note here that I think it's like 6 mummies or 6 human remains that it's based off of, and the strains that came from them. So a low number of remains here. And the story that kind of they were able to draw out is because of the distribution of where those individuals were on the continent and where the range of seals are, they were saying, oh wow, well it's really this particular tuberculosis strain got really far inland, farther than you would expect from just like one spillover event. And so the story goes that this is like some sort of past, right, epidemic or something where the spillover event was just the beginning, and then other stuff happened to have tuberculosis appear so far inland. But of course, with all the caveats that like we're looking at like very ancient single data points and trying to reconstruct what we think.
Yeah, that's kind of demonstrated with this map. We've got like the pinniped. So pinniped means seals, and so they look at like the range of these seals and where they could be found. And I think they initially found a group of like mummies that had these in like area C, and based off of that, they thought, oh, maybe it was just like a spillover event. But then they found some more in area B, and that kind of brought up this idea that, okay, well, maybe this started an outbreak, the spillover event that started the TB outbreak in South America that ended up like raging through the population and then eventually like disappeared before like other diseases could come over that we could study.
Yeah. This is wild. It's such a such a crazy story from the past. Yeah, an interesting.
And also an important warning about seals. So I have it on good authority that you don't want to be bitten by a seal at all because they carry a lot of nasty diseases, and so this is just another example of that.
Another data point.
Don't near seals. Don't bother them. They're dangerous animals, especially the cute ones.
I didn't realize seals have other things that you don't want to get.
Well, I've heard. I mean, I met a person who was working at an Antarctic research station. They were like, "Yeah, seal bites. They're the worst." I'm like, "Okay, well, I'll take your. I'll take." Admittedly, yeah. The viewers of this, you can obviously take this with a pinch of salt because, from your perspective, it's third-hand information from someone who I literally met in a pub. So, I mean.
All right, let's. What do we got
next? Next one is. Microbial regulation of intestinal motility provides resistance against helminth infection.
Oh yeah, this is one of these classic "what does the microbiota do?" questions in papers, but it begins driven from the hypothesis that the response to one of the physiologic responses to helminth, which are worms, intestinal worm parasites. One of the physiological responses in the mammalian digestive system is the idea that you just make a bunch of more mucus and you try to move it out as fast as possible, and that should clear out some of these some of these growing microbes, larvae. The larvae of these worms. Sounds real gross.
Yeah, you got worms, you got poop, and you got like everything. This has got all the gross, this is a gross like kind of like nexus. So
yeah, making more mucus and then moving it more quickly through your body so you can just like clear those worms out the end hole. Like that's the idea behind immunity.
It's
that,
like when
conjuring
you're drinking at the pub and last orders come up and they turn all the lights up and start blaring the worst music you've ever heard just to rush you out. They want you to get rid of you.
Yeah,
it's like that for the human body.
Yeah, time to clear out. And they notice, because it's very fashionable, but it's a great tool to use in mice that get treated with a bunch of antibiotics. They don't really clear out the worms as fast. The response is not the same, and they see the same results mirrored inside of the germ-free condition. So the idea is that when you treat mice with a bunch of antibiotics, it's clearing out their microbiota, and so they want to draw the parallel to this idea that it's microbiota dependent. So they'll verify things inside of germ-free models.
I should explain 3 types of mice used in this study. So there's firstly like nosebiotic, where they've got like no microbiota from birth, and then there's the germ-free where they've like given them there's a germ-free where they have no microbiota since birth. Then there's the antibiotic treated ones where they just clear out all the microbiome, and there's specific pathogen 3, which is basically saying they're healthy mice. They've got no pathogens, but they've got their normal microbiota.
Yeah. And so they'll always want to compare those SPF mice either against the antibiotic treated mice or against the gnotobiotic mice. And both of them are considered this approximation of what happens when you remove the microbiota. But of course, you have to do all of these control experiments to make sure that you're not changing other things through all of this process, because we know that the microbiota is important for a lot of other processes. And so in this paper, they look at some immune signals to say that, okay, but it's not the immune cells that are affecting this particular phenotype. It's something else.
Yeah, and that something else is the intestinal motility. Having a healthy microbiota means that your intestine moves a lot more and has like a healthy amount of mucus. And I think this is something that has been seen in mice that were raised from like early. So I think nosebiotic mice, like not nosebiotic, the germ-free mice, they tend to put on a lot more weight, I think, and they've got less. They're known to have less intestinal motility. But I think this actually like kind of provides a bit more information as to like what that means, and
yeah, they're trying to
yeah,
disentangle it, I think, is one of the tricky parts of the work that they're doing in this paper, because so much is dependent physiologically on the microbiota, it can hit all these different systems. They have to do a bunch of different clever experiments and setups to try to extract the differences from like the greater change to the organism as a whole. One of the ways they do it is they extract the sections of the colon, and they just look at them sort of in an ex vivo scenario where they're just floating in some liquid, and they look at, oh, can they add in certain things to change the peristaltic nature of that? And yeah.
Yeah, I mean, it's really interesting. This is part of because we look at the gut microbiome as like almost like an extension of the human like body, and the question is like, can it form like can it assist the immune system? Like what? What?
Yeah.
I'm all
You all
right.
right?
My apologies to the viewers. I wasn't wearing a mask then, so if you get any symptoms from watching this, then I'm very sorry. Just to clarify, you can't catch coronavirus or like, cold through watching someone who has the cold.
Thanks for clearing that up for us.
I feel like I have to, just in case.
Yeah, so disentangling the effects of the microbiota from the greater, like from the specific effects on the immune system, and then from, I guess, this particular action of gut motility. That I think is the major focus of the experiments in this paper. And then driving down to say that it's this motility actually that is being affected. What affects that specifically in the microbiota? They're not interested in, or they're not trying to address that in this particular experimental setup. But they do offer some potentials, like, oh, it could be some neurotransmitters and things like that. But again, it's a complicated thing to begin to disentangle.
Yeah. Speaking of complicated things sorry, do you have anything else to say on this paper? All right.
Nope. I was going to say that's all I want to say. So tell me.
Complicated things. Identification of bacterial drug-resistant cells by a convolutional neural network in transmission electron microscope images.
Yep. So hello machine learning, this time looking and being trained on electron microscope images of bacteria in order to, and specifically bacteria. It seems like these researchers have worked on a set of bacteria. This is like their little, their playground in some ways, where they had an original strain that then they evolved. They created many variants that are resistant to antibiotics in different ways. And they noticed that even under a light microscope, there are differences between these. And so they apply their machine learning tool set, and they look at pictures that have a lot more features. So they look at electron microscopy. So because that's more zoomed in, you're going to see a lot more different things. Yeah, look at all these images of bacteria. They're being sliced, right? And they're given contrast agents so that become more electron dense. And you get this, the dark parts are the denser parts, I guess.
Yeah. No. This is like feeding in. So, I mean, we can see like what the differences are, but I mean, just looking at this as a non-specialist, I won't be able to characterize what that means. But like, if you run it through like a dispassionate AI, the question is, what will that see what kind of answers will that come up with as to what the differences are, and how can that inform our ideas as to what the differences that are happening in these cells?
Exactly. Yeah. So they use this, deconvolutional neural network. They have like a crazy diagram of how that works. If you people remember when we covered the using AI to discover new antibiotics. I did take a stab at explaining like one of these algorithmic processes in order to analyze what's going on, but I'm not going to attempt to take a stab this time. Essentially, though, at every stage, different features are highlighted from the data, and the computer then correlates those features, right? Like maybe it means like some darker spots over here, or like some weird shape in this region. Those get correlated to whether or not it's antibiotic resistant or not. And they have, right, that's a training set that they have. They already have the two evolved, or in this case, I think they have like 4 different evolved versions of the same bacteria. And once it's trained and they feel like it's working well, you know, there's a lot of parameters that go into that. They then start testing it out, and sure enough, well, they wouldn't be publishing if they found a bad result. You know, one of the many biases in the literature. Sure enough, they're able to discriminate between bacteria that are antibiotic resistant and those that are not. And yeah, in this last figure that you have up, which I find quite interesting, they show us a heat map of the features that are being used to discriminate these bacteria. So they sort of like popped open the hood, if you will, of the algorithm and said, ask the algorithm, show me on this image where you made the decision points. And so you can see the computer is looking at these different areas, synthesizing that information, and that's what it's using to make the decision. And what I find quite fascinating actually about this paper is at the very end, the table 2 below this figure that you have up, they then, because they had like a bunch of bacteria that they evolved to have resistance, they know the genomes of those bacteria, and then they asked the algorithm to compare the gene expression differences in those evolved strains to the features that are being picked out, and they pick up like all these different genes. And what the authors say is kind of interesting about this is these aren't the genes that you would think as being important for antibiotic resistance, like in the mechanisms of antibiotic resistance that we usually talk about. And so maybe it warrants further investigation onto these, what these are doing in terms of being correlated to antibiotic resistance. Or also you could make the statement that this is just like this is just correlated, but there's no mechanistic link, right? Maybe it's like this change plus the other changes that are doing the protection create the visual change that you can see on the slides, something like that.
So yeah, fascinating paper, like the pairing of like computational like AI and looking at microscopy. That's just fascinating, and also the way they try to allow us to look into it or how the algorithm's thinking. Very interesting paper.
Yeah, I like this paper. I'm not really sure like how it impacts like any technologies or like our understanding even of antimicrobial resistance. It is a really interesting to see like the way that this work is being done and how these researchers have partnered, right? Like how they're using computer vision to try to start getting insights on maybe even genetics, right? And genetic regulation. Like there's something here that I found compelling, and which is why I brought it to our attention today.
Yeah. Next paper. A Rhesus channel in the coral symbiosome membrane suggests a novel mechanism to regulate NH3 and CO2 delivery to algal symbionts.
Yeah, I think this is a really straightforward and fun discovery. We're talking about a specific type of protein called the Rhesus channel. It's been identified in a whole bunch of other organisms, and it's known to be able to move nitrogen, ammonium compounds, ammonia across a membrane. Also, other examples is known to move CO2 across membranes. And inside of coral, they have symbiotic algae hanging out inside of the cells. So we know that that algae needs carbon dioxide and nitrogen compounds to survive. And so the question is, are these Rhesus channels the things that might move them across, move those nutrients across the membrane to the symbiotic algae? And yeah, the authors of this paper go out to answer that question. And sure enough, it is. Or yeah, what they can see, from what they see, it does seem like that's the case. That and they even see it at like using super resolution microscopy, I think, again, where they're looking at the membranes themselves and seeing that, I guess, based on like the light cycles or something like that, you see an increase in concentration of these particular transporters in the membrane in order to feed the algae when they
it's
need
interesting.
to be fed.
So like so you look at like day-night cycles as the way to ensure that they can, because the thing that is concerning about like corals at the moment is that sometimes the interaction between the coral and the symbionts can become dysfunctional, where you got these lovely like photosynthetic bacteria, but this bacteria, when it's stressed out, can produce all these reactive oxygen species. These kind of oxygen is great in the doses we usually get, but there are certain versions of oxygen that are chemically quite corrosive. And so when like things that like increases to temperatures happen, sometimes this can cause those photosynthetic like algae to produce toxic substances that can cause things like coral bleaching. So understanding the relationship between these algal symbionts and these corals can give us an insight into how these ecosystems work. And this like specific thing is quite interesting because we're seeing how the coral kind of rations out the resources it gives the algae to prevent it from producing, say, too much ammonium, so like you don't want to give like the algae ammonia, but you don't want to give it too much. You want to make sure it uses that up. Otherwise, you just have this ammonia stacking up. So this is showing how it kind of tapers it off during the night where the algae aren't photosynthesizing. They're not they're not using up the kind of substances that are needed for photosynthesis. Instead, they're using stuff for respiration.
Yeah.
So yeah. So yeah, we can see like in this case, you can see like they pull that protein back away from the symbiosome and back to the host plasma membrane, and then they switch it back during the day.
Yeah. Which is kind of crazy to think at the cellular level, right? Like all this work is being done to maintain a specific biochemical composition in the membranes that, yeah, is going to make your symbiote host happy, or I guess like make them fed at the right times so that they don't, I don't know, create some off products in the times where you don't want them to be active.
Yeah, I think that's a really like interesting relationship, and getting that idea of like these symbiosome membranes, they're not static, they do change throughout the day. And in a lab, we often keep things in very standardized conditions. So this is the kind of thing you need to like have that like kind of, it's very easy for this sort of thing to be ignored in the lab actually. But yeah, having that kind of insight, I think that's very interesting.
Yep. Okay. Moving on, we have population. Oh, the title's eluding me.
Population genomics of Escherichia coli in livestock keeping households across rapidly developing urban landscape.
Thank you. And so they're using E. coli as a marker here just to understand like how do antimicrobial resistance, how do bacteria spread around an environment, and what can that tell us about the spread of antimicrobial resistance? And so, there's many things that are spreading, but for the resolution that they're interested in here is the resolution that they're interested in here is whole genome sequence, because they really want to see like how those genes are all changing across as the strains of bacteria are moving across the population. And specifically, it's also very much focused on an urban environment where there's livestock cohabitating or in close quarters with people. So this is a description in many ways of a specific type of environment and some of the ways in which antibiotic resistance genes could move around in it. And I guess the takeaway would be microbes are everywhere.
Yeah.
What they find is that in general, like they find mixing, a lot of mixing between the E. coli strains on from different households and on different animals, like across Nairobi, and they do say something that likely, so the idea would be that if antibiotic resistance was being selected for in one area, you would quickly see that strain appear sort of all over the place.
Yeah, I mean, this is a very interesting. I mean, because it does it's very in depth and it shows like how different strains kind of diversify and spread. Like it mentions how there are some people working with animals and they get the strains from the animals and they show some similarity, and people within households show similarity in strains. So there is a little bit of the like Orwellian in this, in that you can potentially classify households and interactions based on these shared genomes.
Oh yeah, definitely.
But yeah, so aside from that,
I think this is like something that it's been shown before, like with metagenomics, like with just like sort of shotgun sequencing a whole bunch of things and seeing like sequence identity of stuff. But and in this particular paper, they're doing it on the whole genome scale, so you get even a sense of like within the strains of a single organism how those spread around lots of different places. There's a lot of concepts in here we don't have time to unpack, but one of the ones that I do want to touch on is they talk about this like core genome and accessory genome, because like E. coli, it has like a whole bunch of different genes that aren't in all of the different strains, but then it does have like a core that maintains the same, and they use a combination of the core genome and the accessory genome in order to do these identifications of like where the bacteria are moving. And so again, I haven't read it deep enough to be able to draw out like a specific takeaway from that analysis, but there are very interesting specific takeaways that are brought out by that idea that both the accessories are being swapped around and equally distributed amongst everywhere, as well as the core genomes are being moved around these different households. And the major thing I think that I do want to impart on people that I feel confident in saying is like this is the mechanism in which that we see antibiotic resistance being spread, and so understanding it better and working with this knowledge to create the proper safeguards is important for public health.
Yeah, and I guess that will bring us to the next paper, which is, I think, our final paper: infection strategy and biogeography distinguish cosmopolitan groups of marine jumbo bacteriophages. So this paper had me at jumbo bacteriophages because, yeah, they're big like bacteriophages and they exist all over. And this paper is, I think, essentially they almost like a critique of the scientific literature, because essentially since viruses have been historically considered to be smaller than cells, most of the viral diversity surveys have been geared to the idea that viruses are much smaller than cells. But in recent years, we found that there are megaviruses that are about the same size as cells, or sometimes even bigger, where they almost like impersonating cells in order to infect larger cells. And so, the purpose of this paper was for them to go, okay, look, based on the data now, how would things look like if we actually reoriented how we think about viruses?
Right.
What if we? So they, I think, did a smaller experiment where they. So I mean, one of the examples I give is that a lot of viral surveys rely on plaque assays, which are, you take a lawn of bacteria that are vulnerable to a certain phage. You drop a drop of that phage on it, and the phage should move through the medium. But if the phage is too big, then actually it's very hard for it to move through most media, which are solid. So
okay, wait,
it needs something more liquidy in order to move around.
Wait, I think that's a little bit, that's a bit confused. It's more that they filter the where they think the phages are, and they exclude by size.
Oh yeah,
I don't know if medium.
Yeah, that's another aspect of it. I'm actually talking about literally they're talking about agar concentrations are often too high for larger phage particles to diffuse through compartment compared to small particles.
So, that's
something
that's crazy.
I'd never even thought about.
So just in like the plaque in the overlay, in the agar overlay, they mentioned this.
I think they mentioned this in the introduction. That's where it caught kind of my eye. So this is what prompted them to develop a workflow for changing the way that these bacteriophages are identified by essentially moving the bins for data around.
So they're not repeating like a big survey of the ocean to find these jumbo phages. They have metagenomic data, right, from oceans already, and they're saying, well, let's look for the data features that would show us that there's bigger phages. Essentially, trying to compile, right? Like we mentioned this very early, right? Like in metagenomics, you have lots of all these little short reads. You kind of like combine them into larger and larger pieces, and then you say, okay, this belongs to a single thing. So they're trying to use their knowledge that there's bigger things out there to bin their data in a way that will show them those larger phage genomes.
Yeah, essentially they do that, and they go back to previous data, and they find that phage genomes are generally a lot more widespread than you'd expect. They're found in almost everywhere.
It's pretty wild.
So
I remember we talked about this before, I think, like jumbo phages and how like, oh, back in the day we had a few examples, but more and more examples are coming. Here's like a survey showing like, they're all over the place.
I mean, they're almost like based on like where you look. And that's just fascinating that we've got all this like metagenomic data that has all the secrets in it, that we've got all these like gene data, but we don't necessarily know how to interpret it because we don't have that context of, oh, there are giant like bacteriophages out there. So
right.
So I feel like this kind of like indicates that there is still lots of mysteries in the ocean, and some of them are just sitting in the data that we have.
Yeah, no, that's a really great point. And again, reiterating something that I hope that we're sharing with people is that you only see what you're asking your question for. It's hard to discover things that you don't have some scaffolding to be able to ask the question around. And so, that's one of the major activities of science and of scientific thinking, is being able to formulate new questions so that you can find those new things. Because if you aren't asking the questions, you're not getting the answers.
I think that's a good point to close off the show, actually.
Absolutely. Yeah, so join us next week for more cool microbiology news.
We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can also reach out to us over Twitter at MicroTWJC. We both believe that peer review is this process that we can all participate in. So we hope that you've had a good time listening to us ramble on about microbiology today. If you think you have something to add or find something unclear, let us know. And thank you to everyone in the comments. I feel like it's nice to have discussion between people that are watching us as well.
Yeah, it's a pleasure like reading all your comments and being here with you. And it's been a pleasure chatting.
Same here, Fuzz.
Tune in next week for more microbiology content. Goodbye.